Welcome to this lecture on eCTD Module 3, where we will explore eCTD Module 3, which focuses on chemistry, manufacturing, and controls, commonly referred to as CMC. This module is one of the most critical components of any regulatory submission because it documents everything about how the drug is made and controlled. Today we will cover the structure of the eCTD, dive deep into Module 3 requirements, understand the IND application process, and learn about drug stability studies. The CMC section ensures that the FDA has confidence that the drug can be manufactured consistently and safely. By the end of this lecture, you should understand the key documents required and how they fit together in a regulatory submission. Our learning objectives for today cover 5 key areas. First, we need to understand the overall structure and purpose of eCTD Module 3. This module is where all the chemistry, manufacturing, and controls information lives. Second, we will learn about the investigational new drug application, which is the gateway to conducting clinical trials in humans. Third, we will identify all the departments involved in compiling an eCTD dossier. This is truly a cross-functional effort. Fourth, we will comprehend drug stability concepts and why they matter for regulatory approval. Finally, we will navigate the detailed table of contents for Module 3, understanding the numbering system and what goes where. These objectives will prepare you for working on actual regulatory submissions. The Electronic Common Technical Document, or eCTD, consists of 5 modules. Module 1 is regional and contains administrative information like application forms, proposed indications, and labeling specific to each regulatory authority. Module 2 provides summaries of all the detailed information in Modules 3 through 5, including the Quality Overall Summary, Non-Clinical Overview, and Clinical Overview. Our focus today is Module 3, which represents the chemistry, manufacturing, and controls data. This module contains in-depth information for both the drug substance and drug product. Module 4 contains all non-clinical testing data including pharmacology, pharmacokinetics, and toxicology studies. Module 5 contains clinical trial reports and data. The ICHM-4E guideline specifies exactly how data should be placed in these modules. Let us dive deeper into Module 3. This module is divided into 2 main sections, 3.2.S for drug substance and 3.2.P for drug product. The drug substance section, also called the active pharmaceutical ingredient or API section, covers everything about the active ingredient itself. This includes general information like nomenclature and structure, manufacturing details, characterization data, control procedures, reference standards, container closure systems, and stability data. The drug products section mirrors this structure but focuses on the final dosage form. It covers description and composition, pharmaceutical development, manufacturing, control of excipients, control of the drug product, reference standards, container closure, and stability. Understanding this parallel structure helps you navigate Module 3 efficiently. Compiling an eCTD dossier is a major undertaking that typically takes more than 6 months to complete. Because the dossier represents all the research work done on a product, it requires involvement from multiple departments. Regulatory Affairs leads the effort and ensures compliance with all applicable guidelines. The CMC team provides all the chemistry, manufacturing, and controls documentation. The non-clinical team contributes pharmacology and toxicology data. The clinical team provides clinical study protocols and results. Data management handles the organization and quality of data. Biostatistics provides statistical analyses. Medical writing creates the narrative documents. All these departments are coordinated by the program management team, which ensures timelines are met and dependencies are managed. This cross-functional collaboration is essential for a successful submission. An investigational new drug application, or IND, is a formal request for FDA authorization to administer an investigational drug or biological product to humans.
This authorization must be secured before any interstate shipment and administration of a new drug that is not already approved under a new drug application or biologics license application.
The IND is essentially your permission slip to conduct clinical trials.
Without an approved IND, you cannot legally test your drug in humans in the United States. The IND application demonstrates that the product is reasonably safe for initial human testing and that the proposed clinical studies are properly designed. This applies to both traditional small molecule drugs regulated under the NDA pathway and biologics regulated under the BLA pathway. Filing an IND requires completion of 3 sets of FDA forms. FDA Form 1571 details the study itself, including the study design, objectives, and scope. FDA Form 1572 provides information about the investigators and study sites, essentially a commitment from investigators to conduct the study according to regulations. FDA Form 3674 certifies that the study is registered in clinicaltrials.gov, the national database of clinical trials. Beyond these forms, the IND application must contain information in 3 broad areas. First, animal pharmacology and toxicology studies provide preclinical data to assess whether the product is reasonably safe for initial human testing. Second, manufacturing information demonstrates that the drug can be consistently produced. Third, clinical protocols detail exactly how the human studies will be conducted and by whom. One of the most important aspects of the IND process is understanding the timeline. An IND application goes into effect 30 days after FDA receives it, unless FDA notifies the sponsor that the investigations are subject to a clinical hold. This 30-day review period allows FDA to evaluate the safety of the proposed clinical trials. There are 3 possible outcomes. The most common is simply no notification within 30 days, meaning you can proceed with your clinical trial. Sometimes FDA notifies sponsors earlier that clinical investigations may begin, which can accelerate your timeline. The third possibility is a clinical hold, where FDA identifies safety concerns that must be addressed before trials can proceed. If you receive a clinical hold, you must respond to FDA's concerns and receive clearance before starting any human studies. Once your IND is active, you have ongoing obligations to keep FDA informed. Information amendments are updates to the IND that include essential information not covered by protocol amendments, safety reports, or annual reports. Examples include new toxicology data, updated chemistry information, other technical information, or reports regarding discontinuance of clinical or non-clinical investigations. Annual reports are required within 60 days of the anniversary date that the IND went into effect. These brief progress reports summarize the status of all investigations conducted under the IND, including safety updates, manufacturing changes, and study enrollment status. Maintaining your IND properly is crucial for continued authorization to conduct clinical trials. Drug stability refers to the extent to which a drug substance or product retains, within specified limits and throughout its storage and use period, the same properties and characteristics it possessed at the time of manufacture. Understanding stability is fundamental to CMC. The drug substance, also called the active pharmaceutical ingredient or API, is the material used to manufacture the drug product, usually combined with excipients. The drug product, also called the dosage form or finished product, contains one or more drug substances with excipients in the final packaging intended for marketing. Both drug substances and drug products are typically studied under at least 2 different storage conditions: long-term ambient storage and accelerated conditions. Each stability program is conducted based on an approved protocol. Stability studies on the drug product serve 3 essential purposes. First, they support the stability of drug product used in clinical and non-clinical studies. You need to demonstrate that the material used in your trials remains stable throughout the study duration. Second, stability studies establish commercial expiry dating.
This determines how long the product can be stored and still meet specifications, essentially the shelf life you put on the label. Third, stability studies help determine levels for certain specifications such as API content and preservative levels, and set the control limits for lot release testing. These three purposes directly impact patient safety by ensuring that the drug maintains its quality, efficacy, and safety throughout its intended use period. Stability testing is conducted under specific, controlled conditions. Long-term storage studies are conducted at ambient conditions that reflect the intended storage environment. Typically, this means 25 degrees Celsius plus or minus 2 degrees, and 60% relative humidity plus or minus 5%. These conditions simulate normal storage over the product's shelf life. Accelerated stability studies use higher stress conditions to predict shelf life in shorter timeframes. Typical accelerated conditions are 40 degrees Celsius plus or minus 2 degrees, and 75% relative humidity plus or minus 5%. Testing parameters include physical characteristics like appearance and color, chemical purity and assay, degradation products, moisture content, dissolution or disintegration, microbial limits, and container closure integrity. All testing follows approved stability protocols. The table of contents for Module 3 follows a specific structure defined by ICH guidelines. Section 3 Quality contains the body of data in Section 3.2. The Drug Substance section, 3.2.s, has 7 main subsections: general information, manufacture, characterization, control of drug substance, reference standards or materials, container closure system, and stability. The Drug Product section, 3.2.p, has 8 main subsections: description and composition, pharmaceutical development, manufacture, control of excipients, control of drug product, reference standards or materials, container closure system, and stability. Notice how the structure parallels between drug substance and drug product, making it easier to navigate and ensuring comprehensive coverage of quality requirements. Section 3.2.s.1 covers general information about the drug substance and forms the foundation for all other CMC sections. Subsection 3.2.s.1.1 Nomenclature provides all the names for the drug substance including the International Non-Proprietary Name or INN, the United States Adopted Name or USAN, chemical names, compendial names, code designations, and any other names used. Subsection 3.2.s.1.2 Structure provides the structural formula, molecular weight, stereochemistry, and information about polymorphism. Subsection 3.2.s.1.3 General Properties describes the physical and chemical properties including solubility, pH, pKa, melting point, and optical rotation. These documents provide the basic identity information that regulatory reviewers need to understand your drug substance. Section 3.2.s.2 covers the manufacture of the drug substance and is one of the most detailed sections in Module 3. Subsection 3.2.s.2.1 identifies all manufacturers with their name, address, and specific responsibilities. Subsection 3.2.s.2.2 describes the manufacturing process and process controls in detail. Subsection 3.2.s.2.3 covers control of materials including specifications for starting materials, solvents, reagents, and catalysts. Subsection 3.2.s.2.4 describes controls of critical steps and intermediates with acceptance criteria. Subsection 3.2.s.2.5 covers process validation and evaluation, demonstrating that the process consistently produces material meeting specifications. Subsection 3.2.s.2.6 describes manufacturing process development, including the history of process changes and the rationale behind them. Sections 3.2.s.3 and 3.2.s.4 cover characterization and control of the drug substance. Characterization in 3.2.s.3 includes elucidation of structure, which provides spectroscopic, chromatographic, and other analytical data confirming the structure. This might include NMR, mass spectrometry, infrared spectroscopy, and X-ray crystallography data. Section 3.2.S.3.2 covers impurities, identifying both process-related impurities and degradation products. Control of drug substance in 3.2.S.4 includes 5 subsections. Specification lists all tests and acceptance criteria. Analytical procedures describes how tests are performed. Validation of analytical procedures proves the methods work correctly. Batch analyses provides data from actual batches, and justification of specification explains why limits were set as they were. The drug product section 3.2.P mirrors the structure of the drug substance section. Section 3.2.P.1 describes the drug product including its composition and all components. Section 3.2.P.2 covers pharmaceutical development, explaining how the formulation was developed and why specific excipients were chosen. Section 3.2.P.3 covers manufacture including batch formula, process description, and process controls. Section 3.2.P.4 addresses control of excipients with specifications and analytical procedures. Section 3.2.P.5, control of drug product, is crucial as it includes specifications, analytical procedures, validation, batch analyses, characterization of impurities, and justification of specifications. Section 3.2.P.6 covers reference standards, 3.2.P.7 covers container closure system, and 3.2.P.8 covers stability with summary, protocol, and data. Looking at an actual IND submission, you can see how these sections translate into real documents. In the drug substance folder, you will find the quality overall summary for drug substance, plus individual documents for each subsection like nomenclature, structure, general properties, manufacture, description of manufacturing process, control of materials, controls of critical steps, process validation, manufacturing process development, and elucidation of structure. The drug product folder similarly contains source data documents and individual files for description and composition, components, drug product details, manufacturing process development, container closure, microattributes, compatibility, manufacturers, batch formula, process descriptions, controls, validation, batch analyses, stability summary, and stability data. Each document follows a specific naming convention for easy identification. Let us summarize the key takeaways from today's lecture. eCTD Module 3 is the chemistry, manufacturing, and control section containing comprehensive data on both drug substance and drug product quality. IND applications require FDA authorization before any human testing can begin, and they follow a 30-day review timeline. Multiple departments collaborate over 6 or more months to compile the eCTD dossier, all coordinated by program management. Drug stability studies serve 3 critical purposes: supporting clinical studies, establishing expiry dating, and setting specifications. Section 3.2.S covers drug substance while Section 3.2.P covers drug product, with parallel structures making navigation easier. Finally, IND maintenance requires annual reports within 60 days of the anniversary date and information amendments as needed throughout the development program. Thank you for your attention today. We have covered a lot of ground regarding eCTD Module 3 and the CMC requirements for IND submissions. I encourage you to review the actual Module 3 table of contents and familiarize yourself with the numbering system. Next week, we will continue our exploration of the eCTD by looking at Modules 4 and 5, which cover non-clinical and clinical data respectively. For your assignment this week, please complete the CMC review exercise, which will give you hands-on practice with Module 3 documentation. Are there any questions about today's material? Please feel free to ask about any aspect of eCTD Module 3, the IND process, or drug stability requirements.